Alright, SBI4U, this is video number 6. Here we're talking about advanced photosynthesis. Here we're going to the important thing is to start to understand the electron flow through these photosystems, because it's a lot like the mitochondrial electron flow, but it's a little bit different, and that's often a point of confusion for students. We're going to track oxygen, water, carbon dioxide, CH2O, which is just sugar, and NADPH, which is an energy carrier. It's kind of like NADH. So here's our learning goals. First off, we're going to talk about chlorophyll and the wavelengths of light. That's important because of our keep in mind up there that light has to be a specific energy to hit the electrons. So only certain wavelengths will hit certain molecules, and the chlorophyll is that molecule. Then we're going to get into the electron transport chain in plants. So we call that non-cyclic photophosphorylation, which again is a point of confusion because non-cyclic photophosphorylation, it passes its electrons to the Calvin cycle. So it's non-cyclic because it passes it to a cycle. It's a little bit strange. And then finally we'll talk about cyclic photophosphorylation, which doesn't pass it to the cycle. So that's something to really remember. I'll change the keep in mind to that when we get there. So yeah, without further ado, let's jump into our current topic, which is chlorophyll and energizing the electrons themselves. So I'll take this away, and let's hop in the note. So here's the overview. I'm going to move my face. There we go. Here's the overview. Remember that there's two stages. There's the photo stage and the synthesis stage. So the carbon stuff here in blue is separate from the oxygen stuff over here in red. This makes a bunch of ATP. This also makes an energy carrier called NADPH, which is a lot like NADH. And those bring their energy over to the Calvin cycle, which is taking in CO2 and kicking out sugar. We'll talk about what kind of sugar it's making when we get to the Calvin cycle, and that sugar can get processed into starch and sucrose and all the other stuff that the cell might want to make. So this is the one that takes in light and makes a lot of energy. It happens in the thylakoids, those little structures inside the plant. This happens in the stroma. The stroma is like analogous to the matrix in the mitochondria, but it's that space, the big space inside the chloroplast. So this is going to generate ATP and NADPH. This is going to use ATP and NADPH and send back this spent NADP and ADP and PI to get recharged over here and then come back in here. This is constantly pulling in CO2 from the air. This is constantly kicking out O2 to the air. This is bringing in H2O from the roots, and this is putting out sugar into the veins, and that goes out to the rest of the plant to feed places that can't photosynthesize, right? So like the roots, they can't take in light, or anything like further inside the plant can't take in light, and so it's going to benefit from that sugar that this cycle is making. So these guys aren't just making energy for this cell, they're making energy for a lot of cells in the plant. All right, let's jump in. So first off, there's three main parts to this. There's photoexcitation, which is energizing the electrons with light, and chlorophyll is the molecule that does that. That's why our current topic, chlorophyll and energizing the electrons. Then there's the electron transport chain itself. We're going to jump into that after we talk about this. And then chemiosmosis, which you'll recognize from the mitochondria thing because it's just ATP synthase. So jumping right in, photosystems. Photosystems, the electrons need to get exactly the right amount of energy. And if you look up here, right, light has to be a specific energy to hit those electrons. So the plant wants to make this light, the energy that this light comes in, it wants to make it the right energy to make this electron jump up in energy levels, get the right energy to fall back through the chain, right? Because it has to it starts low energy, then it jumps up, and then it's going to get passed along to a bunch of molecules along the way and do work for the plant. So what this does is every time it hops, it loses a little bit of energy until it's the perfect energy to jump onto this, which is called the reaction center. And it has a molecule, actually two molecules of chlorophyll A right next to each other, surrounded by a bunch of proteins. And it's going to be the final thing to energize this electron, because it is tuned to have exactly the right amount of energy. Like if you look up there, right, light has to have a specific energy. It's tuned to have exactly the right energy to make that electron jump exactly the right level to fall through the chain. So all of these guys are just there to make that energy right. So that way, instead of just one single wavelength of light that hits that center, a bunch of different wavelengths can hit these and pass their energy on. Also, spreading out the antenna complex is more efficient. It's like, you have a big thing up top and the one big protein in the middle, instead of having to make a bunch of big proteins. Right, you just have one here, and light that hits anywhere up here will activate the big protein. So, that's a photosystem. It's got an antenna and a reaction center. The antenna grabs energy and passes it down to the reaction center. Once the energy is the right level, the electron hops. So to zoom in on this, here's the actual molecule that we're interested in. This is chlorophyll. This part here, a lot like a phospholipid, will stick into the membrane. This one here, so see, this is the thylakoid membrane. These guys, the tops of them actually sit on the top of the thylakoid membrane, where they can get the light up on top of the thylakoid membrane. And this is a porphyrin ring. It's a bunch of carbons and nitrogens in this cool like ring within a ring structure. And they are kind of bound by intermolecular forces to this ion of magnesium. We call that a coordinate bond. They're coordinating to this ion of magnesium, and they kind of got it trapped in there. And that magnesium, because it's a positive ion, helps to attract the electrons and helps to transfer the energy to the electron. This is why plants need magnesium in their diet. This is one of the plant's essential nutrients, is actually magnesium from the soil. Now there's two versions of chlorophyll. There's chlorophyll A and chlorophyll B. The only difference between them is in this little carbon here. It's a methyl group on chlorophyll A, and it's an aldehyde group on chlorophyll B. So it's a C double bonded to an oxygen and a hydrogen on the other end for chlorophyll B. And because they're different molecules, they're going to have slightly different energies that they absorb the electrons at, and that's shown on this graph here. I'm going to move my head out of the way. They absorb blue light, and they absorb reddish light. So this is a graph of absorbance. So the big spike is where they absorb the most. You can see the solid line is chlorophyll A, and the dotted line is chlorophyll B. Chlorophyll A absorbs a little bit more in the purple and a little bit more in the orange than chlorophyll B. Chlorophyll B absorbs a lot more in the blue and a lot more in the yellow and less in the orange. And the really cool thing about that is because they're absorbing those colors, they reflect the other colors. The other colors will just bounce right off instead of hitting the electrons, like you see up here, hit the electrons. Instead of hitting the electrons, it just bounces off because it's the wrong energy. And that's why plants are green, because they're absorbing all the blue and absorbing all the red. This is important if you're, buying a light for a fish tank, for example. My fish tank back here has planted, it's a planted tank. It's got real live plants in there, and I have to buy a very expensive light to give them the right kind of wavelengths. There's spikes in these wavelengths so that the plants can be healthy. It's also really cool that because they absorb slightly differently, it changes their color. So down here I've got what's called a chromatograph. Essentially, it was a bunch of just leaf ground up and absorbed onto this paper down here, and they ran probably alcohol or something up, and the different pigments travel different speeds through the paper, and they spread out. This is chlorophyll A up here, this is chlorophyll B. And if you look back here at the chart, chlorophyll B absorbs a lot of blue, but chlorophyll A absorbs a bit more in the violet. And so chlorophyll B doesn't look as blue as chlorophyll A. Chlorophyll A still retains some of that blue, so it's a bluer kind of green. It's a very pretty green. And chlorophyll B absorbs more yellow and leaves orange and reflects more orange. So you can see a little bit of like an orange-ish tone to it compared to chlorophyll A, which doesn't have that orange. So it's that's how the absorbance can change. Even just a little tiny change like that, one atom, right? One atom is changed from a methyl to an aldehyde, and it changes the absorbance by a lot. Right? So yeah, that's chlorophyll. When it absorbs in the right energy, the electron's going to hop up, and that's what we're interested in. That's what sets off the rest of it. There's a question here, why are plants other colors? These are from accessory pigments. You can actually see it back in this slide here. There's some other little accessory pigments. One of them, one common one is carotenoids, because you find them a lot in carrots. They're kind of orange and yellow. Then there's also xanthophylls. The chlorophyll breaks down in the fall because it's a molecule that's built to absorb energy and pass electrons off. So very quickly, once the leaf stops supporting it, it just breaks down. And the xanthophylls and other accessory pigments stick around. The xanthophylls are yellow and orange and stuff because they're absorbing a lot of this purple and blue and green. Yeah, I think it's like to protect the plant. I think it's like almost like a tan for the plant. It protects it from harsh sunlight. Yeah, that finishes our first current topic. I'm going to pause while we get to the next current topic. So, all right, and we're back. So we're talking about non-cyclic photophosphorylation here. Photophosphorylation, if you break down the words, photo for light, phosphorylation is putting a phosphate on something. In this case, we're putting a phosphate on ADP and making ATP. This is kind of a topic that's sometimes confusing for students. So yeah, this is where we're at here right now. This is a topic that's confusing for a lot of people because non-cyclic photophosphorylation, the electrons are passed along the electron transport chain and then they leave. They go to the Calvin cycle. So people get confused. They think that non-cyclic means that it doesn't involve the Calvin cycle, but this is the one that actually does involve the Calvin cycle. It's non-cyclic specifically because it's passing the electrons along and sending it to the Calvin cycle. So just keep that in mind, and that's why it's up in the keep in mind here. The electrons start on water, and then they go to NADPH, an electron carrier that sends it to the Calvin cycle. So I'm going to keep that up there to show us that this is actually going to the Calvin cycle. Here's an overview of the whole process, and I'm going to talk about this slide for a little while. Here in red are the electrons. So this right here, this trace in red, is the electrons traveling through. And this is showing where on the membrane they are, high up outside, almost outside the thylakoid or almost inside the thylakoid. And what I'm going to kind of do here is I'm also going to talk about this in terms of energy, and it kind of shows the energy level just because the way they travel from inside to outside is kind of like their energy level, but yeah, it's not exactly their energy level. Then there's also the hydrogen that comes through here, so there's kind of a annoying thing about this diagram is they've used the same color for the hydrogen movement as they have for the electron movement, but I'm sure we can figure that one out. So let's go through the whole electron transport chain in the mitochondria. The goal here is to start on H2O, and finish on NADPH. That NADPH is going to go to the Calvin cycle, take the electrons to the Calvin cycle. Just like NADH brings electrons in to the mitochondrial cycle, here it takes it away. Now in the mitochondria, the electrons end up on H2O because it's a low energy molecule, and the H2O will leave. But here the H2O comes in from the roots, drops off its electrons, and those are low energy electrons. They can't really do anything, but the plant has the photons. It has the antenna complex. It can take those electrons and kick them up in energy, and then they can start falling through the chain and doing things. So the thing that kicks up the electrons first is called photosystem 2. That's annoying. Also a common mistake people make. They say photosystem 1 does it first. No, photosystem 2 is the first one in the chain. It's called photosystem 2 because they discovered it second, and they just kept that mistake the whole time. It's also often called P680, and the 680 is the light that it absorbs. The reaction center there, the two chlorophyll A molecules that are right next to each other, they absorb at 680 nanometers. Photosystem 1, which we'll see later, which also kicks electrons up in energy, absorbs at 700 nanometers. It also has just two molecules of chlorophyll A, but the proteins around them are different, so it changes the absorbance a tiny bit. Photosystem 2 has chlorophyll A and B in it. Photosystem 1 is mostly chlorophyll A, just a small molecular difference between them. I'm not going to ask it on a test, but just interesting to note. So the electrons come off of water. They're low energy electrons. They get kicked up by photosystem 2. They get high energy. Then it gets passed along to an electron shuttle. Just like we saw electron shuttles in the mitochondria, we have electron shuttles in the plant. This first one's called plastiquinone. It grabs the electrons. It also grabs some hydrogens, and then it passes the electrons off to the B6F complex, and the hydrogens fall in at the same time. This B6F. So this is kind of like your proton pump. Notice that there's no other proton pumping action happening on this electron transfer chain. There's only one pump in the chloroplast, but it's a very good one. It pumps 4 electrons when 2 electrons go through it. If you remember with mitochondria, there were 3 separate pumps, and each one pumped just 1 hydrogen when 2 electrons went across. So this one's actually more efficient. 4 hydrogens for every 2 electrons instead of 3 for the mitochondria. B6F takes the electrons, passes it down, and passes it to plastocyanin, which is another electron shuttle, a small molecule that brings the electrons from here to here. Then the plastocyanin passes it off to another photosystem. This is now photosystem 1. Photosystem 1 kicks up the electrons in energy again by pulling in sunlight. They get kicked up in energy. They get passed on to yet another electron shuttle called ferredoxin. Then the ferredoxin brings it to the last thing, NADP reductase, which is reducing NADP. Right? Reduction is a gain of electrons, and the NADP is gaining electrons, turning into NADPH, and it's going to head off to the Calvin cycle to bring those electrons and that energy into the Calvin cycle to help make sugar. So it's acyclic, it's non-cyclic because it's moving from water off into the Calvin cycle. So that's what the electrons are doing. Let's look at the protons. The protons are getting pushed in. There are also protons coming off of the water because the water is going to split into hydrogen and O2. The O2 is just a horrible toxic byproduct. Off it goes. The plant spits it out through the leaves and transpires it through transpiration. The H plus joins a bunch of other H plus, and you've got a big buildup of H plus inside, not a lot of H plus outside, and they want to fall through ATP synthase and make ATP. Well, they want to turn ADP plus P into ATP. Then they kind of float back around and get shoved back in. They're also getting cleared by the NADPH. The NADPH is pulling H pluses out of this solution and sending it to the Calvin cycle. So this is reducing the amount of H plus out here. Both of these are increasing the amount of H plus out here, and you get yourself a gradient, and you can make ATP. That ATP also goes to the Calvin cycle, and you can also use it to, the plant cell can also use it for other things. It's just normal ATP. That is non-cyclic photophosphorylation. The photo part refers to the electrons gaining energy from light. The phosphorylation part happens down here with ATP synthase, same molecule as the mitochondrial ATP synthase. That is acyclic or non-cyclic photophosphorylation. Put that away. A couple little keep in minds here. ATP is generated from reactions from photosystem 2 here. Photosystem 2, right, it gives them the energy to fall down through B6F and push the protons across. And ADPH is generated from the energy from photosystem 1, but it needed the energy from photosystem 2 to get there. Right? It can't do this on its own. It needs a source of electrons, which are coming through photosystem 2. So it needs a source of electrons. Yeah, that's non-cyclic photophosphorylation. So you need to know what photophosphorylation is and why this is non-cyclic. That's up there and down here. Here's an actual energy diagram here, where you can see that it kind of climbs up in energy, falls down, climbs up again, falls down. There's still high energy here because the Calvin cycle wants that energy to make sugar. And then more water needs to come in for this to continue. This is why the plant needs water through the roots and why it'll die if you don't water it. Can't make sugar. Now we're going to get into the Calvin cycle. I'm going to fix up the keep in mind and stuff, so I'll be back in 2 seconds. And I'm back. Here's the Calvin cycle. We're going to go through the Calvin cycle really quickly. I'm not going to spend too long on it, especially because there's a video down below here that I implore you to watch. It is an excellent, excellent, excellent video about the Calvin cycle, much better than I could explain it. Keep in mind up here that the Calvin cycle keeps carbons in circulation, and those are split between many molecules. It's a difference between it and the Krebs cycle. The Krebs cycle only has, it's 4 carbons in circulation most of the time, and it's just on one molecule. And it just runs a couple of times, once for every acetyl-CoA. Here you're actually going to see a lot of molecules, and I'll get into that. So this is also sometimes called C3 photosynthesis because the first compound has 3 carbons, and it pulls in CO2. It pushes out sugar. That means that it requires energy. Remember our first law of thermodynamics that if something releases energy one way, it must require energy the other way. Taking sugar and turning it into CO2 like we do for energy releases energy. So taking CO2 and turning it into sugar must require an input of energy. So this is going to take ATP and an ADPH. This happens in the stroma. Here's a diagram of it. The stroma is that space inside the chloroplast, kind of like the matrix of a mitochondria. Here's the cycle. That's a really kind of rough view of it because it's not sharp, but we're going to break it down into individual steps. What's important here is for the overall is that 3 CO2s are going to come in. They come in one at a time, and 1 G3P is going to come out. And if G3P sounds familiar, that was the molecule in the middle of glycolysis, right? So sugar, you have to put in energy to get G3P, and then the G3P, you release energy as you come down to pyruvate. It's the G3P. It's a high energy sugar precursor. It can make glucose, it can make fructose, it can make sucrose and cellulose, all of those guys that plants make. And because it's a high energy one, it's actually kind of free to make glucose with it because a glucose molecule is lower energy than 2 G3Ps. You just fall down. So 3 CO2s come in, 1 G3P comes out. It takes ATP in, it takes NADPH, so that's the energy, and some more ATP over here. That all gets made by the thylakoid. So let's talk about the first part. This is called carbon fixation. You might hear carbon fixation In other contexts, it just means taking carbon dioxide and fixing it, turning it into some kind of useful compound. So carbon fixation. 3 CO2s come in. There's this, it joins, I don't really have what it joins. It joins ribulose, where is it, RUBP, ribulose bisphosphate. That's a 5-carbon sugar. It's a 5-carbon sugar. So one of them joins to this 5-carbon sugar. It makes a 6-carbon thing. That 6-carbon thing very quickly splits into two of these 3 phosphoglycerates, 3 PGAs. And that happens 3 times, but really it's 3 rounds through the cycle. Yeah, so this is a 5-carbon. A 1-carbon comes in, makes a 6-carbon. That thing very quickly gets turned into the 3 PGA. When you have 3 come in, it'll make 3 of these 6-carbon intermediates because it's going to join with 3 of the 5-carbon predecessors, and then those 3 6-carbon intermediates are going to turn to 6 3-carbon molecules. Yeah. This joining is done by an enzyme called Rubisco, which is ribulose 1,5-bisphosphate carboxylase oxygenase, Rubisco, and it will carboxylate it, which means adding the carbon dioxide, but sometimes it does the wrong thing and oxygenates it instead. And so plants make a lot of this protein just to make sure that they have enough where it will carboxylate. And it's the most abundant protein on Earth because it's helping the plants make sugar. And the plants making sugar is the food source for everything else. So it is the number 1 protein on Earth. Yeah, so 3 CO2 plus 3 of these ribulose bisphosphates, they turn into this intermediate, then they turn into 3PG. The 3PG goes through some processing. It takes the ATP, the energy from the ATP, gets some extra phosphates. See how it has like 1 phosphate here and then has a 2nd phosphate here? That came off the ATP. There's 6 of those, so it needs 6 ATP. The 6 of these then need to, kind of get some extra hydrogens and stuff, and they pull the hydrogens off of an ADPH. This also gives them some energy that turns them into G3P. Hey, G3P, that's what the cycle makes. But it makes 6, and it only pulls out 1 out of the 6, because if you remember up here, 15 of them are going to stay in circulation. So after this step, the 15 of them are going to stay in circulation, and one 3-carbon molecule gets pulled out, which means the three of them are going to get pulled out for every 3 CO2 in. Then the G3P goes through a series of steps where it's kind of reshuffled into the 5 G3Ps get reshuffled into 3 ribulose bisphosphates. So it's 5 3-carbon molecules get turned into 3 5-carbon molecules, which are still 15 carbons. That takes some ATP to do, because it needs a 2nd phosphate to be ribulose bisphosphate. And the cycle continues. It just keeps going and going and going. So to summarize, CO2 comes in and joins a 5-carbon thing, turns into 2 3-carbon things, and then every kind of 3 times through the cycle, 1 G3P comes out. But the other 5 G3P keep on going through, and that's the Calvin cycle. Pause again, we'll go to cyclic photophosphorylation. Okay, I'm back off pause, and this is the last piece. This is cyclic photophosphorylation. This is the last piece here, cyclic photophosphorylation. It's a photophosphorylation, so you know it's going to make ATP, but it's cyclic, so it doesn't go to the Calvin cycle. Once again, we've got to watch out for that point of confusion. It's cyclic, so it's cyclic because, as you can see, the electrons are moving in a cycle. They just keep going around and around and around and around. So you don't need water, no water. and you don't need NADPH, or you don't make NADPH. So this can't go to the Calvin cycle because there's no NADPH. The Calvin cycle needs NADPH. So we're going to zoom in here. This is done on photosystem 1. Photosystem 1, you see the photons hitting it, jumping around, hitting the reaction center, and they go up to the primary acceptor on the reaction center, and where normally they would pass to ferredoxin and go to NADP reductase, they pass to ferredoxin, just like normal, but then the ferredoxin passes it to B6F instead. And B6F is earlier in the chain. Then the B6F will push it down, push hydrogens through, and it'll make ATP. And then it'll pass on to plastocyanin, and then back to photosystem 1, and then it can go back up and get back another kick in energy from a different photon, up to the primary acceptor, back through ferredoxin, and repeat the loop over and over and over again. So these electrons are just shoveling through over and over again. And every time they pass through B6F, I'm going to jump back through the slides a little bit here to that one. Every time they pass through B6F, 4 hydrogens come through. So they're coming through this system, going to plastocyanin, getting kicked up in energy, passing the ferredoxin, but then the ferredoxin just drops them back off over here, and then they pass through again. Drops them back over here, they pass through again. And every time they pass through, 4 hydrogens. You're building up lots and lots of proton gradient. You can make lots and lots of ATP. And because it's a cycle, just like it says up here, right, just a cycle, you don't need water because you don't need new electrons. You've got old electrons going through the cycle. And you're not making NADPH. You're not, you're not helping the Calvin cycle make sugars, but you are making a bunch of ATP, which is great for the plant anyway. It's great for the plant cell to do other things besides just sugar. Besides just using sugar. So that's cyclic photophosphorylation, and just the electrons moving in a loop, making lots and lots and lots of ATP. The plant can do this, indefinitely. There's nothing, as long as it's got ADP to turn into ATP, it can do this whenever the sun is shining, and it does. More advanced plants, stuff like grass and flowers and trees and things like that, they have both of these systems built in, both the cyclic and the acyclic. Simpler stuff like blue-green algae, they just use the cyclic photophosphorylation and make the ATP, and just use the ATP. They're not really making the sugar using the photosystem 1. Yeah. And only ATP is made, no NADPH is made. Similarities between the two, both make ATP. And even the acyclic makes ATP. Both take energy from light. 1 photon makes 4 ATP for both. Both use electrons as the energy carrier. They both use photosystem 1, and that means that they also use the electron shuttles for photosystem 1, the ferredoxin and the plastocyanin. They also both use the B6F complex, but all the other ones are only in acyclic. There's a big long list of cyclic and acyclic. I'm not going to read it to you because, why would I? That's the end of the content there, but I'm just going to highlight 2 things that are going to be down in the description. The first one is that video, Nature's Smallest Factory. Please watch it. It'll give you a really nice understanding of the Calvin cycle in very, very simple terms. It's also like Dr. Seuss kind of characters. Very, very cool. And the other thing is, if you want to try this, you can actually do this leaf chromatography thing. This thing. You can actually do that at home with some alcohol and some coffee filters. It's not going to look nearly as good as this, but you might be able to separate chlorophyll A and B and get a really nice picture of it. The keratin comes out really, really early too, so you can actually get that yellow color separate from the green. It's kind of neat. So try that one out if you want to give it a go. Yeah, that's the end of the lesson. Let's just review our learning goals. We talked about the wavelengths of light and why they're important. We talked about the electron transport chain in plants and how it's similar and different to the electron transport chain in mitochondria. Basically it's backwards, and there's more electron carrier, or more electron shuttles, but there's only one pump, but that pump is really, really good. And it can be backwards because it's got the photosystems kicking it up. Photosystem 2 first, photosystem 1 second, because biology clings to history that way. We talked about the Calvin cycle with its 15 carbons going in a circle, 3 CO2s coming in, and 1 G3P coming out to make glucose and various other things. And then we talked about the cyclic photophosphorylation, that neat trick that plants use to just make a bunch of energy literally for free, because all they need to do, all that needs to happen is the sun is shining. One last piece, maybe one last keep in mind, and I'm just going to write it really big, and I'm going to pause the video while I do it. Okay, I've unpaused, and I'm going to bring you a very special message, and that is that plants also have mitochondria. Why would plants possibly also have mitochondria? Mitochondria take sugar and they make energy with oxygen, and the plant has lots of sugar. And the chloroplast can only make ATP when the sun is shining. So here's some reasons plants might want mitochondria. Nighttime, sun's not shining, gotta make energy, use your mitochondria, use that sugar that you make during the day. Or maybe it's winter, right? You're covered in snow and you still gotta, make sugar. Or you're growing in a seed in the ground and you can't get light. You need mitochondria to get the sugar that you've stored up in this nice seed that your mom plant gave you. Or maybe you're deeper in the plant, like the heartwood of a tree. You're surrounded by bark, you can't get any sugar. Or you're getting sugar from the leaves that are bringing it to you. Or the roots are underground. They need to grow and repair and all this stuff. They're getting sugar from the leaves, and they're using their mitochondria to process it. So please remember that plants do also have mitochondria. So that's it. Thanks a lot, and I'll see you in class.